Cell's replication is essential to all parts of life, whether it be single-celled or multicellular. Cell division in bacteria is an important process as it ensures survival of the species. However, bacteria do not have sex. Instead, they take one cell and divide into two. Today we're going to see the process and how bacteria replicate, as well as able to make an exact copy of DNA. Prokaryote replication. What is replication?
Bacteria
Replication is when
reproduce
one
very
cell
simply
becomes
and
large enough to
rapidly
split
by
into
doubling
two
their
identical
contents and
cells.
splitting in two.
This
Just one
ensures the survival of
bacterium,
the species or
dividing every
organism.
20 minutes, could produce nearly 5,000
How do prokaryotes
billion...
replicate? Binary fission. All prokaryotes perform binary fission, where one cell will split into two cells. The new cells will be able to split again and again and again, increasing the numbers.
Almost all bacteria cells reproduce by binary fission. As
A prokaryote will replicate its DNA and important proteins and change into two new cells. It's almost identical except for mutations. Prokaryotes, through binary fission, you can replicate into many, starting with one cell. They will replicate the DNA and then split into two new cells as shown in the following process. This can keep going and going and going until bacteria are able to reach their carrying capacity. How does DNA replicate? Each new cell needs an exact copy of DNA in order to function. Don't forget, DNA is the important molecule that dictates everything the cell to do. So if you're going to have two new cells, you need to have two new strands of DNA. Uses a process called semi-conservative replication. Step 1, the original DNA strand gets unzipped. In this diagram here, we see a protein binding to the DNA and causing it to unzip, allowing us to have two separate fragments. These fragments can be read, and identical copies can be made.
Using DNA polymerase.
DNA polymerase will then bind the DNA and begin to read each strand. In our diagram here, we see one of the strands of DNA has some proteins attached to allow DNA polymerase to attach. It then creates a matching base thanks to the base pairings that go with DNA.
The process repeats.
Due to base pairings, it can match up the correct bases to create two identical strands. Here we see the protein binding once again, and thanks to the magic of base pairings, every A will have a T on the other side, and every G will have a C on the other side. This ensures that both strands are going to be identical. Each strand goes into a new cell.
The genetic information in a bacterial cell is stored in the form of a double-stranded, covalently closed circle of DNA.
We'll have a strand of the original DNA as well. Here's another way of looking at it using this bacterial chromosome. In this bacterial chromosome, we have one side of the DNA labeled the blue and the other side labeled the red. In the semi-conservative process of DNA replication, the strands will separate from each other.
This is referred to as semi-conservative.
Once the strands have fully separated, they're lacking the other side of the DNA necessary to function. They need the matching base pairs in order to be able to work. DNA polymerase will then come through and make the matching strands. Notice, one red and one blue, indicating that each one had a strand of the original
Eukaryotic cells, which are our cells, replicate very differently.
How does a mutation occur? Sometimes a mistake could occur from reading DNA too fast.
We still need to replicate our DNA. So semi-conservative replication in DNA still occurs. So if that's the case, what's the process that our cells, eukaryotic cells, replicate? Think about it, and I'll see you guys when you come back from the break. Have a good one.